Hello, my name is Kelsey Holt, and today I will be discussing a knockout discovery, otherwise known as gene targeting in mice. This groundbreaking research was done by Mario Capecci, Martin Evans, and Oliver Smithies. Before we begin, I would like to briefly review some vocabulary concepts you may not be familiar with. First, we have knockout mouse. A knockout mouse is a mouse in which a gene has been inactivated. This can be done by placing artificial DNA into the genome. The artificial DNA replaces the real DNA, causing the gene to become inactive or knocked out. I will be going into more depth on this later in my presentation. Next is homologous recombination. Homologous recombination is the exchange of DNA sequences between chromosomes. As you can see in the image here, the two chromosomes have overlapped and then split off, creating two new sequences of DNA. Next is reporter genes. Reporter genes can be used to track the location and function of genes. The reporter gene is turned on if the target gene turns on. It is sent into the nucleus with the artificial DNA. In order to create a knockout mouse, researchers alter a harvested embryonic stem cell. These stem cells are injected into embryos. The embryos are then placed in a foster mother and carried to term. The resulting offspring are either chimeral or mosaic mice or wild-type mice. The chimeral mice are bred with normal mice in order to get the new gene into the germline. The offspring of the chimeral mice are heterozygous for the desired trait. These mice can then be bred to create mice that are homozygous for the desired trait. You're probably wondering how researchers alter the genes that they put into the embryonic stem cells. There are two popular methods, homologous recombination and gene trapping. The first method I will discuss is homologous recombination. In homologous recombination, embryonic stem cells are used. A gene in the nucleus is altered by introducing an artificial piece of DNA. The artificial DNA is recognized at the cell's nucleus and is swapped in place of the real gene. The artificial gene is inactivated and causes the true gene to be knocked out. This diagram from Macmillan Higher Ed shows the steps of homologous recombination very well. In this case, the vector used is a plasmid. The targeted gene is inactivated and then inserted into a mouse stem cell. The targeted gene lines up with the gene in the mouse genome, and recombination occurs. The inactive version of the gene is now in the genome of the mouse, and the normal gene is lost. The stem cell is transplanted into the mouse embryo, and once the mouse is born, it is examined to see if it has the inactivated gene. The second method is called gene trapping. In this case, artificial DNA containing a reporter gene is inserted into a gene at random. The cell's RNA is unable to properly perform splicing, preventing the original gene from producing its protein and limiting the function of the gene. This again knocks out the gene. This diagram from the University of Manitoba depicts a basic gene trap vector. The vector is comprised of a reporter gene, a splice acceptor, and the desired marker. It is added to the ES cells either through viral infection or electroporation. Electroporation is a process that uses electricity to push substances, in this case the new gene, through cell membranes. It does this by creating pores in the cell membrane. Due to the introduction of the gene, RNA activity is disrupted, and it causes the new marker, in this case a neo-R gene, to be produced. There are advantages to both types of knockout. With homologous recombination, researchers have a little bit more precision with the gene knockout. With gene trapping, researchers don't need to know specific DNA sequences. This means a single vector can make a lot of different generations of mice with knocked out genes. The discovery of knockout mice started with preliminary research on homologous recombination, which I discussed earlier. Now, I'm going to discuss each researcher's contribution to the research that led to the Nobel Prize. First up is Mario Capecci. He was born in Verona, Italy in 1937. He spent his childhood in the streets of Italy after his mother was taken to a concentration camp. When his mother was finally freed, they reunited and moved to the United States. He studied at Antioch College, where he received a bachelor's degree. He received his PhD from Harvard before joining the faculty. He then moved to the University of Utah 4 years later to do his research. In the 1980s, he used recombinant DNA to inject DNA into the nucleus of cells. This enhanced gene transfer. Using Evans and Smithy's work, he created knockout mice. Among his awards are a Kyoto Prize, a Franklin Medal, and in 2001 he won the Lasker Award. In 2002, he won the Wolf Prize in Medicine. Capecci began by injecting DNA into the nucleus of cells using small glass needles. The apparatus he used is shown here. In 1977, Wigler and Axel developed a process to inject TK-positive DNA into cells. However, it was ineffective and had a very low success rate. Capecci believed he could have more accuracy with the injection process he used, and he was right. He found that to make the transfer as efficient as possible, the gene must be linked to a viral DNA sequence. When a virus was linked to the gene, the TK-positive cells were generated with much higher frequency. The virus was a transmission vector carrying the new DNA into the cell. Capecci found that even though multiple copies of the HSV gene could be inserted randomly at different places into the cell nucleus, they would all be found at a single locus. This can be seen in the image here. He and his colleagues determined that this was due to the work of homologous recombination. He believed if they could use this homologous recombination between an introduced DNA molecule and an original DNA molecule, they would be able to manipulate the genome however they wished. Through studying homologous recombination, he and his colleagues realized that linear DNA was the most effective type of DNA to use in order to achieve high production of the desired cell types. Capecchi wanted to see if defective genes could be repaired through this DNA implantation technique. He wanted to study the effects of this homologous recombination on resistance to the drug G418. In order to study this, he and his colleagues injected a cell with a dysfunctional neo-R gene with a copy of a gene that had a 5' point mutation, indicated by the asterisk on this diagram. About 1 in 1,000 cells were corrected by the new gene inserted into the nucleus and developed resistance to the drug G418, as seen by the neo-R positive gene in the third chromosome on the bottom. Now Capecchi had to figure out how to apply this, not only in cells, but in living mice. Next, I will discuss the contributions of Oliver Smithies. Oliver Smithies was born in Yorkshire. He received his master's and doctorate in biochemistry from the University of Oxford. He then studied at the University of Wisconsin. He moved to the University of Toronto, where he developed the starch gel electrophoresis technique. In 1988, he moved to UNC Chapel Hill, where he did his research. He used Evans' work to show that removal or alteration of genes allowed the genome of mice to be manipulated. He and Capecchi worked together to breed mice with specific conditions. In 1991, he made a knockout mouse with cystic fibrosis. His awards include the Lasker Award for Basic Medical Research, a Wolf Prize in Medicine, the NC Award for Science, the O. Max Gardner Award, which is the highest award for faculty in the UNC system, and the American Institute of Chemists Gold Medal. In his free time, he was a pilot, despite being colorblind. Smithies' research was similar, but independent to that of Capecchi's. Capecchi himself stated in a lecture that they both focused on gene targeting, and at times were a bit competitive. Smithies' first major contribution to science was starch gel electrophoresis, which allowed proteins to be separated much more efficiently than ever before. With this starch gel electrophoresis, Smithies discovered blood proteins, known as haptoglobins. While studying haptoglobins and hemoglobins, Smithies became interested in homologous recombination. Show his original plans for the starch gel electrophoresis. He said he was reminded of the way his mother's boiled starch cooled down after she had used it to do laundry when he was a child. Smithies wanted to figure out a way to replace the gene that leads to sickle cell anemia with a functional cell. He was able to use homologous recombination in order to insert the gene into the mammalian genome. Smithies was also able to create the first animal model for the study of cystic fibrosis. His research showed a possibility of being able to disrupt genes in the mammal genome, which then led to the advancement of Capecchi's work. They worked together with Evans to develop the first knockout mouse. The third and final researcher who contributed to the development of knockout mice was Martin Evans. Evans was born in 1941 in Gloucestershire, England. He studied at the University of Cambridge and received his bachelor's and master's in biochemistry. He received his PhD from University College in London in 1969. In 1978, he joined the faculty of Cambridge University, where he performed the majority of his research. In 1999, he accepted a post at Cardiff University. In 1981, he discovered embryonic stem cells in mice. He realized that these cells could be used to transmit genetic material. He used embryonic stem cells to create mice that had less Schneehan syndrome. These experiments led to the development of knockout mice. Among the awards and honors he received are the Lasker Award, a knighthood, the Gold Medal for the Royal Society of Medicine, and the Copley Medal of Royal Medicine. Knockout mice likely never would have been possible without the work of Martin Evans. Until this point, the delivery system of altered DNA into mice was not perfected. Evans was researching a solution. Evans' main line of research was embryonic carcinoma cells. He was trying to deliver genetic information via these cells. However, they proved ineffective due to their abnormal chromosomes. This meant that they were unable to contribute to germ cell formation. He discovered that normal cells could be taken from mouse embryos, and he named these embryonic stem cells, or ES cells. These cells could become almost any kind of tissue. The picture shows differentiation of stem cells. Embryos from one mouse strain were injected with the embryonic stem cells that had differentiated. These embryos were then carried by foster mothers. The pups were born, the gene was present in the chimera offspring, and the chimera were bred in order to ensure the new genes would be inherited. Evans actually invited Capecchi and his wife to his lab and taught them how to derive mouse embryonic stem cells and how to produce the chimera mice by adding altered embryonic stem cells to mouse embryos. Evans used retroviruses in order to integrate the genes into the chromosomes. Retroviral DNA from the embryonic stem cells was transferred to the mosaic mice into the germline. In 1989, the first embryonic stem cells were used to create knockout mice. All three of these researchers' separate discoveries combined to allow the advancement and discovery of the technology that made knockout mice possible today. There are many uses of knockout mice. Mice have a very similar genome to humans, making them great to experiment on. It allowed researchers to study how genes present in mice can affect humans. Common uses of knockout mice are to study cancer, obesity, heart disease, arthritis, substance abuse, anxiety, aging, and Parkinson's disease. There are numerous uses for knockout mice, and hopefully one day researchers will be able to study the entire genome. Here I will discuss knockout mice being used in breast cancer research and obesity research. Knockout mice are commonly used to study breast cancer. One gene that has been identified in playing a role in breast cancer is the p53 gene, which is a tumor-suppressing gene. Mutation in this gene can lead to Li-Fraumeni syndrome. Women who suffer from LFS often have higher incidences of breast cancer. Also, in people who have mutations of the BRCA1 and BRCA2 genes, high rates of mutation are found in the p53 gene. The picture indicates mice that have tumors, though these are not necessarily the mice that were used for this experiment. Researchers found that when the p53 gene was knocked out, mice succumbed to other tumor types, such as lymphomas, before breast cancer could be studied. This led to a problem that they needed to solve. Researchers decided to use tissue-specific deletion to alter the gene. This inserts the desired gene, however the gene does not recombine until a recombinase is expressed. This figure shows some of the functions of p53. When p53 is activated, it aids in apoptosis and cellular protection in order to prevent tumorigenesis. When further investigated at the cellular level, it was found that the p53 gene had very important apoptosis effects. Mice with damaged p53 genes had lower rates of tumor suppression than those with a functional p53 gene. Mice were exposed to radiation in order to study the effects of exogenous stress on tumor growth. It was determined that the p53 gene actually played an instrumental role when combined with the effects of the BRCA1 gene. 0% of mice expressing the genotype BRCA plus minus and p53 plus plus develop mammary tumors as compared to 10% of BRCA plus minus and p53 minus minus mice. This shows that BRCA1 and p53 play a cooperative role in breast cancer development. At first, BRCA2 research showed that the BRCA2 mutation was embryonically lethal. However, researchers eventually found a strain that was able to survive the mutation. In mice with a defective BRCA2 gene, deficiency of just one p53 allele was enough to cause tumor growth in mice. It was also found that in human patients who have BRCA1 and BRCA2 type breast cancers, there are high rates of p53 mutation, which indicates an important role in p53 in tumor suppression. p53 plays a role in tumor biology, and deficiency of it promotes aggressiveness, rapid growth, and genetic instability in mammary gland tumors. Researchers believe that p53 mice will help lead to more studies surrounding the development of breast cancer by determining more pathways that p53 cooperates with. It is hoped that the p53 gene can be studied more in order to increase our understanding of the development of breast cancer and eventually its prevention and treatment. I will now be looking at a specific study of obesity using knockout mice. There have been many studies performed studying obesity using knockout mice. Some genes studied are the MC4R, KSR2, SIM1, and BDNF genes. All have been found to be correlated with obesity. In order to study obesity in humans, researchers knocked out the G2E3 and the PRKD1 genes. G2E3 was knocked out via gene trapping, whereas the PRKD1 gene was knocked out via homologous recombination. The mice shown here suffer from obesity due to having a knocked out gene. While it is not the same gene that was used in this research, I think it is helpful to see what these mice may look like. This image shows where scientists knocked out the G2E3 gene in the chromosome. Mice were fed the same diet and cared for in the same way. It was found that in the G2E3 knockout mice, there was a marked increase in body weight compared to their wild-type counterparts. Researchers found an 11% increase in body weight, a 65% increase in body fat, and a 48% increase in body fat percentage. It was also found that the G2E3 knockout mice were glucose intolerant. PRKD1 mice, on the other hand, had comparable levels of body fat as compared to their wild-type counterparts. The above chart shows the results of the study. Researchers decided to study the G2E3 gene because it plays an important role in the UPS system, which is a protein regulatory system in cells that allows for protein homeostasis and function. Researchers concluded that obesity-related SNPs located near G2E3 gene decreases the amount of functional G2E3 protein, thus leading to an increased risk of obesity. Researchers are studying obesity because it has become a major public health issue. Researchers wish to identify genes responsible for obesity and eventually provide therapies for obesity prevention and treatment. With the use of knockout mice, researchers can effectively study the genome and isolate obesity genes, which will hopefully lead to the prevention of obesity in the future. Thanks to the work of Capecchi, Evans, and Smith, we will be able to uncover many scientific breakthroughs pertaining to public health. It will be exciting to see what the future of knockout mice holds. My references and all the sources I cited. Thank you so much for watching. I hope you enjoyed learning about knockout mice.